Hello everyone, welcome back to Dopamine Biology. Please make sure to leave a like and a comment. Do not forget to subscribe. Today we will be talking about how 2 meters of DNA fit into a cell nucleus. When scientists first decided to sequence the human genome, it seemed an impossibly large and complicated challenge. A decade since achieving this aim, scientists are faced with a similarly overwhelming challenge: understanding the folding of a human's entire genetic data into a tiny cell nucleus. The folding of the genome influences not only which genes are transcribed, but also internal wiring of the genes themselves. The human genome consists of deoxyribonucleic acid, or DNA, the genetic material for all living organisms. Each person's genome resides in a cell's nucleus in the form of chromosomes and is a sequence of around 3 billion nucleotides comprising around 30,000 genes, which dictate the instructions for creating and maintaining a human being. There are 4 different DNA nucleotides, each defined by a specific nitrogenous base: adenine, thymine, guanine, and cytosine. Stretched out, the DNA molecule is about 2 meters long and must be carefully folded and compressed into a complex three-dimensional structure, which does not tie itself in knots and fits within the nucleus of the cell. To express our genetic instructions, the gene sequence is copied or transcribed into a ribonucleic acid or RNA molecule, which travels out of the nucleus into the cytoplasm and is translated into a final protein product. The genome's three-dimensional structure can help decide which genes are expressed and which are not. The nucleus is partitioned into regions that are busy and active, and where many genes are transcribed, and other compressed regions where genes are silent. By presenting a gene into an active region, the genome structure can influence which genes are transcribed, which genes are not. Human genes are made up of small parts called exons, which are separated by non-coding DNA sequences called introns. When a gene is transcribed, the intervening introns are cut or spliced out, and the exons are strung back together to form the sequence that is then translated into protein. By including or removing different exons, the same gene can be spliced together in different combinations that are then translated into different protein products. When we examine the three-dimensional genome structure, we will see that within a gene, exon sequences are often folded together while the intervening introns are looped out. Human genome contains a total of 6 billion base pairs of DNA per cell. Because each base pair is around 0.34 nanometers long, each diploid cell therefore contains about 2 meters of DNA. Moreover, it is estimated that the human body contains about 50 trillion cells, which works out to 100 trillion meters of DNA per human. Each of us has enough DNA to go from here to the sun and back more than 300 times, or around Earth 2.5 million times. How is this possible? The answer to this question lies in the fact that certain proteins compact chromosomal DNA into microscopic space of the eukaryotic nucleus. These proteins are called histones, and the resulting DNA protein complex is called chromatin. Chromosomal DNA is packaged inside microscopic nuclei with the help of histones. These are positively charged proteins that strongly adhere to negatively charged DNA and form complexes called nucleosomes. Nucleosomes fold up to form a 30 nanometer chromatin fiber, which forms loops averaging 300 nanometers in length. The 300 nanometer fibers are compressed and folded to produce a 200 nanometer wide fiber, which is tightly coiled into the chromatid of a chromosome. Thanks for watching.